Okay, thank you. I want to talk about the Great Prairie Project, which is one of the largest ecosystems in the U.S. This first slide indicates the Wisconsin native prairie site. And in the Midwest, the prairies are virtually all remnant prairies, meaning that there's some reason that in the 1800s somebody didn't plow them up, like they had too many rocks or were sandy or something like that. So we have chosen these prairies now managed by some conservation reserve and a matched long-term agricultural site. So in the same soil type, same other conditions. So it's matched to the prairie. And we have these sites in Wisconsin, Iowa, and Kansas. So the rationale for this. So the Midwest Prairie represents the largest expanse of the world's most fertile soils. It's a reference site for understanding biological basis of ecosystem services of microbial communities. Since a lot of work has been focused on the ocean, this is the comparable terrestrial system. The ocean's largest gyres that represent important carbon sequestration and biogeochemical cycling sites, as does the prairie ecosystem in North America. It sequesters the most carbon of any soil system in the U.S., and of course it produces a large amount of biomass annually, meaning plant biomass as well as microbial biomass. It also represented a technical challenge because soil is considered the most diverse site for microbial communities and the genes they carry. So these are the sampling sites. The sites in Wisconsin, in Iowa, and in Kansas. And they represent a transect across the prairie, going from colder to warmer, and as you go west, drier sites. The design is indicated here. So there is a reference core taken up in this corner here, and that is the sample that was used for metagenomic sequencing. Then next to it, immediately next to it, 1 centimeter away, was another core at 1 meter and at 10 meters in each direction. So these were sequenced for 16S ribosomal genes by pyrotagging to see how representative this core was of the area. So what questions can we answer? Of course, the major overlying question is, how does this very different land management, 100 years of cultivated agriculture, affect the microbial community, its gene content, compared to the native prairie, never been cultivated? So we can ask questions at various levels, such as the population level, the gene level, and particularly important is what I call the ecofunctional genes. Those genes that directly drive the ecosystem processes of importance. And then eventually down to the expression level. Can spatial scale of the habitat be related to gene diversity? Because at some level, the microbial community is not diverse. It would be microcolonies of a single genotype. So how can we relate that small scale to gene diversity? What is the effect of restoration? In this case, native prairie grasses and switchgrass on diversity. So the Wisconsin site has 2 additional sites adjacent to the continuous cultivated site, which is a restored prairie and switchgrass. And then what are the spatial patterns of populations of genes, and are they altered by different land management? So who lives in the Great Prairie soil based on 16S ribosomal sequencing? And so this is a phylum level evaluation from the pyrotag information. And you see it's dominated by proteobacteria. Second, acidobacteria, the group for which very few organisms have been isolated and only a few genome sequenced. But in general represent about 1/3 of the microbial biomass of terrestrial temperate soils at least. Actinobacteria, and then outward. So that's who lives there in terms of the phyla. Do the microbial communities differ among the sites? And the answer is yes. These are color-coded by the various sites. So the yellow and the green and the purple are the native prairie sites, but they are different somewhat from each other. These are the corn sites on this, or the long-term cultivated sites over here. I should say that at sampling time, All of the long-term cultivation was in corn, all sampled at the same period, all sampled in June, the same period of the year. If we merge that information together in the color coding, so this is all the native prairie here, and this is the corn here, and in between is the switchgrass and the restored prairie. So previously, these sites were also continuous agriculture, and they map more closely with the agricultural sites, but maybe beginning to move towards the native prairie community types. And for the next slide, remember that green is always the native prairie, and red is the cultivated agriculture. So these are some potential indicators of management practice. So green, as I indicated, is prairie. So these are alpha proteobacteria, and these are acetobacteria. And the size of the dot indicates the extent of those materials, or those representatives in the sequence. So the alpha proteobacteria and the acetobacteria were certainly more selected by the prairie than the other management conditions. This is an example that Gina pulled out that was interesting because it's bradyrhizobium. And bradyrhizobium, of course, is the symbiotic nitrogen fixer associated with soybeans. This, however, is all native prairie. Never had a soybean in it. And yet the alpha proteobacteria, including the bradyrhizobium, are more dominant there than they are in the cultivated sites, some of which I know have had considerable soybean production. What about the effect of restoration that I mentioned before? Well, here is the native prairie again, the agriculture, and then the sites that have been, at the time of sampling, only 2 years in the grasses. You saw the sampling design, some samples close together and then getting more distant. Do we see a spatial pattern? And the answer is no. These are the distances shown here. It's hard to read where those are here, but maybe you can see the red bars. The red bars indicate no statistical difference between those locations. So the summary point here is that sampling did not affect, or the community structure was not recognizable as different at this sampling scale. So that would suggest that the metagenome perhaps is more representative of this area than the small sample core from which it was originally taken. So what's the sequencing status? So the original plan was to sequence about 50 gigabases per site. And this would be the Iowa sites, the Kansas, the Wisconsin, and then here would be the 2 new restored grass sites. But then HiSeq became available, so we also tried HiSeq, so that added a substantial amount of sequence to that. The Iowa set is highlighted here because work that we've done at Michigan State has emphasized this comparison, and some of the work done at Lawrence Berkeley is emphasizing the Kansas comparison. As I think most people recognize, the soil environment is very diverse. What is this diversity challenge? And based on K-mer recapture analysis done by a person in Titus Brown's lab, this is sort of the estimate. So for the Iowa corn at 136 gigabases, the estimated coverage is 0.7x. The prairie at 62 gigabases, 0.82x, to give some indication. And then if we compare that, for example, to reach the rumen depth of sampling of the top abundance organisms, we would need 1 to 2 terabase of sequences from these samples. At 5x coverage, it's about 1 terabase. Going forward to 10x coverage, up to 2 terabase, and then we would get less than 1% novel sequence, just as a calculated perspective of where we might be on coverage. So what are the obstacles related to assembly? So one is the scalability of the assembly approaches, which, as they have been done, simply won't work at this scale. The high diversity in soil. the non-uniform coverage of organisms or representation of organismal groups in the community, the availability of computational resources to the broader scientific community, and we lack reference genomes, much different than the Human Microbiome Project. So what can we do with too much data? And Titus Brown at Michigan State is a very clever guy in computer science. And so if we deal with this problem, he took the approach of data partitioning as illustrated here. So not to partition the soil, but to partition the DNA from the soil into different components, then that make the problem manageable at a smaller scale. So these are the two data reduction techniques that one can consider. So one is to eliminate reads that do not connect to many other reads, or to group reads by connectivity into different partitions of the entire graph. And so this is Titus Brown here with Sparty. And this is the approach then showing the different colors, which would be the different partition bins. So one of the problems that appeared is indicated here, and that is as you get more sequence, you get less assembly. And that is because then you begin to get artifacts in linkages because of errors in the Illumina sequencing. So the more you sequence, the more you get an error that will connect it to something that already exists. So here is the low density assembly graph. Here's a higher density assembly graph illustrating this problem. So the artifacts cause preferential attachment, as I indicated. So this indicates those points there that I indicated. But it's mainly caused by the Illumina errors. And so the solution to this problem that Titus came up with is illustrated here. So basically, to trim before you get these additional errors. So as you go in this direction, when you begin to get these different linkages, then trim before that particular point, and therefore correct for these artifacts. Okay, so with that in place, then what can we assemble? So if we take the Iowa corn data, HiSeq plus the GA2 reads, a 219 gig base of sequence, and we assemble that, then we can get 148,000 contigs. So the cutoff here was 1,000 base pairs. 220 megabases, maximum length of 20,000 bases, maximum coverage 10x. This was all done on the Amazon EC2 for 1 week for under $500. So it makes the problem more manageable and more cost effective. So what can we do once we get those assembled sequences? So this was put in the camera analysis pipeline. We could identify 344,000 ORFs, this being the longest, this being the shortest, and this being the average ORF, and this would be the kind of annotations that came from that analysis of what was assembled. So that's where we are now in demonstrating that approach and what the first level of things that you can do with that. In the 16S sequences in the Iowa corn, we noticed two groups of organisms that were more dominant. And so we sequenced the genomes that were isolated from those groups. One was Frituria and the other was Saprospira. So those were sequenced. So if we use those as reference genomes and compare our assembled data to those, this is the number of hits that we get to the amino acids. And then if we compare all of the short reads to those references, we get these kinds of numbers. And in the end, about 1.5% of the Frituria draft genome gave 5x coverage. Now, I would point out that this was a Frituria strain, I think, that was in the German culture collection. no idea of how it really might relate, how closely it might relate to the species or the groups that were found in the Iowa soil. So this outcome might be a reasonable one, given that. So summary of the assembly partitioning approach is this. So the exact approach for scaling the metagenome assembly would work at the 10 to 100x scale, feeds into the existing assemblers, particularly velvet, retains low abundance reads, so nothing is thrown out if there is a single match. Successfully applied to 200 gigabases of the high-seq data from the Iowa corn, and run entirely on a single Amazon rental node with 68 gigabases of RAM. The partitioning approach also helps resolve the genome mixture issues because, like reads, always go to the same bin. So that's the assembly portion, necessitated by the very short reads of Illumina, which are too short to get to the gene level. But of course, there's a lot of sequence that doesn't assemble. So I think it's very important to have a program that deals with short sequences and what one can learn there. And I'm particularly interested in what I call the ecofunctional genes, targeting those, because those are the ones that drive the important ecosystem processes. I'm less concerned about the 50% of the genes that are in the core that drive central metabolism, because we know they must be there, and they're not going to tell us much about that particular environment. So what are the approaches that we can use? And we've done several here. So the first is to find a method to detect a model functional gene. And the one we've started with is nifi, important to nitrogen fixation. The database is reasonable for it in GenBank, and so much is known about it already. So it represents the model to try various approaches. And so one thing we tried is illustrated here. The gene is about, actually, generally about 880 bases. And this is information then on the nifi background, sensitivity, specificity, false discovery rates. So the first is to evaluate what is feasible in searching for this particular gene. So we use two approaches, the hammer approach and the Kamer-based approach is indicated here. And I should also indicate, and I forgot earlier, that the pictures here indicate the people who did the work on the various portions of the work I'm talking about. So we took 199 taxonomically diverse genomes that contain nifi. There were 168 nifi genes identified in those 109 genomes. 166 were annotated, 2 were unannotated but highly similar. And then we generated reads from the genome sequences. So we basically cut that up as if it was a metagenomic mixture, and then evaluated the outcome from that, as indicated here, translated it into the 6 protein reading frames. And then evaluated the Kamer approach, which is shown here, and the Hummer search, as indicated here, according to sensitivity and specificity in an ROC curve. So the good, where you want to be is in the upper left-hand corner here. So at this level of evaluation, the Kamer performed better, but it's also an exact search. So in real life, it might not perform so well compared to the HMM model. Okay, so what did we find? So if we look at the 1.1 billion reads from the Iowa Corn and Prairie, from the Hummer nifi search, we found 35,000 reads. However, most of those reads map to the N-terminus of that nifi model, which is not very specific for nifi. There are lots of other genes that are proteins that have that motif. So that region is non-specific to nifi. However, with this reduced number, We could blast those. 524 had top NIF-H blast hits. That would correspond to 1 NIF-H gene per 370 bacteria in soil, assuming an average genome size and population of bacteria in soil, which I think is a reasonable number that one might expect in a soil environment. This is how that would predicted to be perform over the number of sequences we have and the bit save scored. So it's feasible. The point is it's feasible. Now, Rachel McElprang at Lawrence Berkeley Lab then has been using the nucleating assembler, which came from this group working on the Rumen project. And this illustrates that point here. And to me, it's genome walking from a computational point of view. So this was started with a blast to find the original set of NIF-H sequences, though it could be the Hummer output that I just showed you. And then a certain sets are pulled out. They represent the starting contig. And then from that, one then looks for the overlapping ends, as indicated here, then extends that contig, and repeats this. I think she repeated this 28 times. And in the end, you get a computationally assembled gene. So that NIF-H was not found in the global assembly that she did previously on the 50 gigabases, not the one that I talked about from the partitioning approach, but this was one that we did that was done previously. We covered 10 full to nearly full-length genes, and they were from actinobacteria, alpha proteobacteria, and beta proteobacteria, where you might expect these organisms to be found. And from what we found before, the alpha proteobacteria were particularly dominant in these environments and have NIF-H genes. So the method is extendable to larger data sets and multiple genes. So where we are now is with this proof of principle and several approaches, then to begin to extend it to other genes. Another approach that relates to this gene-targeted approach is amplicon sequencing. We call it gene-targeted sequencing. And again, of NIF-H in this example. This was done from over 1 million sequences from 4 geographic regions. And then if we compare those to what is in GenBank, we find that most of them compare to isolates already known in GenBank. And if we look through what those are, many of them are alpha proteobacteria. And if I go farther down, many of them are bradyrhizobium, matching the native prairie result that I showed before. Some delta proteobacteria, other groups, beta proteobacteria, and so forth. In this particular, and we have a reference set here, so we've taken all of the NIF-H sequences from GenBank, clustered those, made a stable reference group, and of about 992 reference sets that we can compare other sample data to. But most of what we find is 95% amino acid identity to the closest isolates already known. So this, in general, is a surprising result. And we have done this kind of work for other sequences like aromatic degrading genes important in the carbon cycle, and it's completely different there. Over half of what we find in every sample is not represented in GenBank. So there's a different range of outcomes, I think, in these functional genes. Okay, then in closing, what's next? So the big theme here was land use change, 100 years of cultivated agriculture versus the native ecosystem. What's next on this scale? And to me, on the terrestrial side, it's the Amazon, because it represents the world's largest CO2 sequestering ecosystem. Very important in controlling atmospheric gases on the global scale. And of course, there has been a tremendous loss of native forest in that region. I'm involved with several of the people indicated here in a microbial observatory project in Rondônia State in Brazil, in which land use change on the microbial communities is being evaluated. And just to indicate a similar kind of sampling design here, though it's at a much larger scale, 100 meters here, and in the region, then out to 10 kilometers, replicated out to 10 kilometers. So at a larger geographic scale. And we can learn some interesting ecological points. So this is the distance decay relationship. In other words, how communities vary in similarity as you go out in distance. So this is what you would often find, and in the primary forest, that relationship holds. But the conversion in this region is primarily to pasture land and beef production. So this is a pasture converted in 1987, and you see the distance decay relationship is almost non-existent. So that would be one ecological change. So the land use change does not reduce the number of taxa, but it does alter composition. So this would be the number of taxa which are similar. This is the change here. So these are pastures formed at different periods of time, and then a successional forest at the end. And you see that there is a change in different groups over time, but the forest returns to the original type. And this figure is using the geochip done by Zhizhong Zhao, or people in this group working in Zhizhong Zhao's lab, to evaluate the number of functional genes. And this is the prairie converted in 1911, 1972, 2004, the primary forest and the secondary forest. The point here is the number of functional genes in the original forest and the secondary forest are similar to each other, where they are reduced in the prairie, is indicated there. Okay, so in conclusion, so soil is diverse? That's the question. And the answer can be no, or it can be yes, because it depends on the scale sampled. So if you sample, as I indicated before, a microcolony on the surface of a clay particle, you will have a pure culture. And there will be no diversity. The problem is, of course, that we don't sample at that scale. We sample at a larger scale, and then we begin to experience this kind of environment in soil. And the point I make here is that soil actually is a composite of many communities and a complex of subhabitats. But it's also a very old environment. This type of environment here, without the macroorganisms, has been here since for the last 3.8 billion years. In more recent times, we have higher organisms that also contribute to the community. But it's a very old community with lots of different environments within, lots of different gradients. That creates diversity. That also then raises the sampling issue, at what scale we sample. And of course, when we sample at a larger scale, sequencing centers always want extra amounts of DNA, so you've got to sample at a larger scale. So finally, to acknowledge the groups that have worked on this so far. So at MSU, Titus Brown is a key person because he's a very clever guy from the computational side. And the postdoc that he and I share, Dina Howe. Jim Cole has also been a really important contributor, and Jordan Fish, an important guru at coding. And then at Lawrence Berkeley and at JGI, a number of people have contributed, but the ones highlighted here are the ones that have been major contributors in the work so far. And then at the sites, these are primary people at each of the sites that we've studied that helped us with the sampling and collecting all of the metadata. So thank you, and any questions?
Questions?
So do you ever think about mixing all your prairie soils and assembling it as a group?
Oh, mixing all the data together?
Yeah, right.
So definitely, that's one. So that's one's question at a different scale. You know what? So the DNA taken for sequencing was actually taken from one core, but also one aggregate from that core. So that's a small scale. So then we can ask, in the same environment at a different location, what do we add in terms of gene content? Yeah. I guess, one point, I guess, on the previous slide that I forgot to make is this in the last corner. How important is assembly? I mean, is assembly only something that's a carryover from the history of genomics when we had one genome and, of course, we wanted to assemble it? Because in an environment like this, it may be the gene content that's much more important than the particular organism that it resides in. It may reside in a particular organism by chance, actually. And so assembly beyond a gene level may be not so important.
Anything else? Okay, well, thank you very much. Sarah.